afternoon. Today's mini lesson is going to be on nucleic acids. So nucleic acids are the polymers that are made up by nucleotides. Nucleotides are just the monomers of nucleic acids as the polymers. So nucleotides are those building blocks that we stick together to make nucleic acid. So they do have super important functions, and they function to store, transmit, and express hereditary information. So hereditary information is just information that is passed on from a parent to a child. So it does come in two forms. It comes in the form of DNA, or deoxyribose nucleic acid, and ribonucleic acid, or RNA. And they do have different structures, so therefore they do have different functions. So what are the components of a nucleic acid? So we start with our nucleotide, and then we put a bunch of nucleotides together and we get polynucleotides. And then we put even more nucleotides together and we get a nucleic acid. So nucleotides have three parts. They have a nitrogenous base, a 5-carbon pentose sugar, and a phosphate group. So this is only seen in polynucleotides. Each monomer only has one phosphate group. So we do have different nitrogenous bases. They can be classified into two types, pyrimidines and purines. Pyrimidines have one ring with 6 atoms around them. So it has these are our cysteines, cytosines, thymine, and uracil. And then our pyrimidines, or purines, which have one ring with 6 atoms bonded to a 5-atom ring. And this is going to be adenine and guanine. So it is really, really, really important to remember that thymine is only in DNA. And we can tell thymine and uracil apart because thymine has an extra carbon hanging out right there. Uracil is only found in RNA. Oh, it's supposed to be a highlighter. Only found in RNA. And so we are going to have an pyrimidines and a purines bond together to start making polynucleotides. So the 5-carbon sugar is a pentose sugar. So if you remember back in geometry, we talked about pentose shapes, and this is a pentose carbon. So it's a sugar is bonded to the base. In sugar, it's a deoxyribose, in DNA, it's a deoxyribose. In RNA, it's a ribose sugar. So you can tell the different sugars because RNA, once again, has an extra oxygen, while DNA is missing that oxygen here. So since they differ in structure, they are going to differ in function. The phosphate group is added on to that 5-carbon sugar, which then is attached to the base, and that forms a nucleotide. So it is a big, big, big compound. So we can tell this is deoxyribose because it doesn't have that oxygen right there. So that phosphate group is going to bind to that ribose sugar, and that ribose sugar is going to bind to the nitrogenous base. We can tell it's a nitrogenous base because we've got those nitrogens there. And we can tell it's a phosphate group because there's a phosphate there. So once we start binding them together, we're going to get these polynucleotides. So these are phosphate groups that are linked to adjacent nucleotides. So they are going to start linking to adjacent ones. They link directionally, so they're going to link in one way, just like our amino acids. And we read these 5 to 3 prime. That's really important to remember. So you can see the sugar phosphate backbone, and then the nitrogenous bases are just sticking off to the side. Quick, tell me, is this one right here a pyrimidine or a purine? And then which one is this one right here? Pyrimidine or a purine? Phosphate in. So you see the 5 to 3, and most of the time we are going to write those on there. The sequence of the base along the DNA or the mRNA is unique for each gene. So it is super unique because the structure is different than the function. And a gene is a piece of that genetic hereditary information. So it then tells the amino acid what to become. So it dictates the amino acid sequence and then dictates the structures of the proteins. So remember, structure dictates function. This is a great example of it. So DNA consists of 2 polynucleotides. So 2 of those long chains coming together. And they form a double helix bond. So these strands are anti-parallel, so they go against each other. One goes one way while the other goes the other. But they are held together by hydrogen bonds between the bases. So we can see it here in our little image. They are anti-parallel, so there's a rule, and this is a super important rule that I want you to remember. It's called Chagoff's rule. And that is that adenine always pairs to thymine and guanine always pairs to cytosine. It's called Chagoff's rule. It's super, super important to remember. So, RNA is just a single strand of polynucleotides. They vary in shape and size, and this is due to the base pairings between them. Now, remember, structure dictates function, so RNA's structure is different than DNA because adenine binds with uracil instead of thymine, and cytosine still binds to guanine. So, I want to give you some time to practice. If we give you a segment of DNA that reads like this, we know that they are anti-parallel. What is the other side of the DNA? Right? So you can see that guanine always pairs to cytosine, adenine to thymine, so on and so forth. Great. Now let's work on some more practice problems. Great. I will see you later in the lipid lesson. All right, so today we are going to talk about lipids, and this will be a mini lesson on lipids today.